In the last 3 lectures, we covered microbes of the rumen. In the following 3 lectures, we'll complete our survey of microbes of the gastrointestinal tract by discussing microbes of the ruminant hindgut, humans, and other species. The current lecture will talk about microbes of the ruminant hindgut. By the end of these 3 lectures, you should be able to contrast the gut physiology and microbiology of the rumen and ruminant hindgut, as well as ruminants, humans, horses, and other species. For these 3 lectures, there is no reading from the textbook. It does not discuss these topics. Peter van Soest commented that the lower part of the gastrointestinal tract receives much less attention than the rumen. One would think that ruminants had no lower tract. Today we'll discuss what little is known about the ruminant hindgut. The hindgut is variably developed across ruminants. We saw these 2 figures before. The dik-dik, the small African antelope for example, has only a very small cecum. The sheep, by contrast, has a very large cecum, and quite a bit of digestion can occur there. This table summarizes the contribution of the rumen and the hindgut to degradation of substrate, production of short-chain fatty acids, and other items. For cellulose degradation, for example, the rumen contributes to 70 to 90% of the total, whereas 10 to 30% of it occurs post-ruminally. For hemicellulose digestion, the rumen is responsible for 60 to 100% of the total, and 0 to 40% occurs post-ruminally. That is to say that less cellulose and hemicellulose degradation occurs in the hindgut, but it's not negligible. The short-chain fatty acid concentrations in the rumen and the hindgut are surprisingly similar, which solidifies that fermentation is occurring in the hindgut. The contribution of short-chain fatty acids to maintenance energy is much higher from the rumen than in the hindgut, however. In the rumen, it's 30 to 80%. In the hindgut, it's only 5 to 7%. And this is attributable to the cecum being much smaller than the rumen. There are 2 items for which the rumen alone is important, and that's microbial protein and B vitamins. As we mentioned before, microbial protein synthesized in the rumen contributes to 50 to 90% of the metabolizable protein requirements of the animal. In the cecum and colon, it's 0% because microbial protein cannot be absorbed. The same is for B vitamins. 100% of the ruminant requirement is supplied by the rumen microbes. B vitamins cannot be absorbed in the cecum and colon. In the hindgut, microbial groups present are bacteria, methanogens, flagellate protozoa, and fungi, just as in the rumen. The ciliate protozoa, however, are absent. Viruses are probably present, but they have not been reported. There are some cultured groups of bacteria that overlap in the hindgut and the rumen. The Prevotella, Lactobacillus, Butavibrio, Ruminococcus, Streptococcus, and Selenomonas species are found in both sites. There are some groups that are found only in the hindgut and not in high concentrations in the rumen. For example, the Bifidobacterium, Fusobacterium, and Micrococcus species. Methanogens and fungi are present in the hindgut. The methanogens are Methanobrevibacter species, as in the rumen. Similarly, the fungi are the same genera as in the rumen. The abundance and activity of these groups is available in just a few studies. It's available for bacteria and methanogens. It's not available at all for other groups. We'll now review some of the few studies that are available. This study compared bacterial abundance between the rumen and cecum in sheep. It compared both total counts and cultured counts. We can see for total counts that the values were slightly higher in the rumen versus the cecum for most sheep. For cultured counts, values were much higher in the rumen versus the cecum. There was usually a 10-fold difference. This suggests that there may be fewer live bacteria in the cecum versus the rumen, though it could be that they're uncultured for some other reason. A more recent study compared bacterial abundance using molecular techniques, specifically RNA probes. Here the authors compared the abundance at various times after feeding. They saw that the abundance in the rumen was higher than in the cecum for most time points for total bacteria. The cellulitic bacteria, when expressed as a percentage of total bacterial RNA, did not differ much between the rumen and cecum. It was about 5%. Looking at individual species, Ruminococcus alvis, Ruminococcus flavifaciens, and Fibrobacter succinogenes, we see nearly equal abundance in the rumen versus the cecum. This study compared the abundance of bacteria as well as methanogens using genes that are marker for those two groups. We see that both methanogens and bacteria were in nearly equal concentrations in the rumen and cecum for two different diets, wheat and corn. The data that we've examined to this point were for abundance alone. These data show fibrolytic activity in the rumen and the cecum at various times after feeding. The authors found that xylanase activity, that is, activity towards a purified xylan, was much higher in the rumen versus the cecum, tenfold higher approximately. Avicillase activity is that activity towards avicil, a purified cellulose. It was relatively high in the rumen, but it was almost negligible in the cecum for most time points. These substrates are purified, and they may not resemble that of the native feed, but these are the data that are available. The study that measured methanogen abundance also measured methane production after adding either wheat or corn in vitro batch culture. And we see that both for wheat and corn substrates, that methane production was much higher for rumen contents than for cecal contents. So this seems to suggest that methanogen activity is higher in the rumen versus the cecum. For this lecture, the take-home messages are that the hindgut is less important versus the rumen for fiber digestion and short-chain fatty acid production, but the contributions still are not negligible. Ciliate protozoa are absent from the hindgut, but groups are otherwise similar, at least at a rough scale. The hindgut likely has lower microbial abundance, and it very likely has lower activity, both towards fiber and for methane production, but more comparisons are needed.